everyone. Welcome to Biology 1124. If you're watching this, you should be in the online sections, section 39 and section 40. My name is Shannon Reeves, and I will be your TA for this semester, and I'm going to take you through this PowerPoint. So I am here in the lab. I have the whiteboards and the television set up behind me just in case there's any principles that we need to go over that might require me to use the whiteboard. Last semester, I just did screenshots of me going over the PowerPoints and there was no video of me, and I figured maybe this could add some sort of personal touch. We've noticed that it's hard to get to know the students online given all this COVID stuff, and wanting to reach out to your teachers online is not something students are wanting to do. In my own experience, it's not something I particularly want to do either. So at least maybe you can see my face more often so that if you do have questions, you feel compelled to ask them to me and to email me or meet in person if you would like to. So again, the coordinator for all of the labs is Dr. Shaw, Dr. Taryn Shaw, and he is the one who puts up everything on Canvas and decides the labs that you will be doing. I'm just the facilitator for teaching you the weekly labs. So my name is Shannon Reeves. If you are ever in lecture and they ask you what your TA's name is, it is Shannon. This is my email address down here, and my office hours will be Tuesday from 12 to 2 and Friday from 12 to 2. You can either meet in person with me, which for some students is a real plus to be able to meet in person because they're visual learners, and you may have questions on what the labs have to do with lecture, and being able to draw chromosomes and mitosis and meiosis instead of using your hands may be super necessary if you're a visual learner. But if you feel like meeting over Zoom is something you want to do, or if you have, if you're immunocompromised in some way, then feel free to socially meet over Zoom. But if these times don't work for you, remember that I am available at other times, so we can work something out to meet either in person or over Zoom outside of these hours as well. Just email me. So I study epidemiology. In the previous picture, there's just a picture of me, a headshot. I use that in my in-person labs because I have a mask on all day and they don't actually know what I look like. And also because Dr. Shaw encourages us to use pictures of us in the field doing our work so that people know what we study. But I study epidemiology, not fish or birds or something really cool where you can take an action shot. I study data. So my action shots are really lame, so I just had a picture of myself. But I study epidemiology, so the official definition is study and analysis of distribution, namely who, what, and where, the patterns and determinants of health and disease conditions in defined populations. And so that definition is a mouthful, and really what it means is that we are scientists that work in the medical community. But instead of working on individuals and their health, we work on populations to see what diseases and health determinants are going in and out of certain populations. So those of you that are in maybe pre-med or HES may be in clinical health over here. So clinical health is about individuals, not necessarily about populations as a whole. Clinical health deals with diagnosing people, treating people, procedures, stuff like that. But public health is the realm really that epidemiologists and biostatisticians are under. So public health is more about the population's health as a whole. That's why widespread, escalating, and serious health issues such as COVID-19 are considered public health crises. So you all are probably more familiar now with epidemiologists than you were a year ago. Most of the time, whenever I got asked what epidemiology was, somebody would say something along the lines of skin. I do not study skin. I study epidemics, epidemiology, or endemics or pandemics. So things that you'll see with public health are ideas more like, and these are all things that I've studied in the past, comparison of opioid use among Native Americans and rural and metropolitan Oklahoma cities. So in Native American populations, opioid addiction is a really big issue out here in Oklahoma. And so knowing whether it's a bigger issue in rural places or metropolitan places helps to know how professionals, medical professionals, and public health professionals can um, make policies and change and direct efforts and money and resources towards whether there's more issues in rural areas or metropolitan. There's also increasings of sexually transmitted diseases among Uganda refugee women, um, preference for medical marijuana over maybe opioids for medication among HIV/AIDS patients in the US. This was mostly done so that um, those who have HIV and AIDS due to needle sharing are not tempted to go back to opioids. They can use medical marijuana as pain treatment. Or maybe if you have a geographical comparison of use of biological weapons across the world and who's using ancient DNA as biological weapons versus who's using good old normal biological weapons. This differs because obviously clinical health, you're going to a doctor, you're going to a PA or a nurse practitioner or somebody, maybe a physical therapist, and they're treating you. So they're diagnosing you. They would maybe treat you with cognitive behavioral therapy if it's depression or mental health that you're going to get assessed for, physical therapy, nutrition, that's all clinical health. So the data that we use deals with people as populations as a whole so that we can figure out as a whole where we can put our efforts. It's really cool, so if you have any interest in looking into it, feel free to email me. So here's my action shot. The coolest thing I have is that I got vaccinated since I work with COVID patients. So right now what I'm currently working on is misinformation during COVID. So how social media affects how people see news, and then therefore how the coronavirus moves through populations that use social media as news instead of maybe news or scientific data as news. And then the other one would be the impact of cancer treatment on COVID-19 patients among Native American populations in Oklahoma. So this is really cool research that I'm interested in. If you have questions about COVID, I'm knee-deep in research for it. Feel free to ask. If you have questions about biology, I'm here too, though. So this is Ravioli. She's my assistant professor. She's very helpful. She's a little bit judgmental, but she has a good heart. So if she appears on the slide, then it is because that is important information that you will need probably not only for this investigation or this week's assignment, but future ones as well. So even though we are in lab and typically we don't think about having, a notebook or a folder for lab, I strongly encourage you to have one, especially since you're doing things online and you won't be seeing me from week to week. But Ravioli, I'm opposite. Ravioli will help. So what you should have done so far, last week we had an assignment for the week of 125. This is what your module will look like if you go into Canvas. You should have learned how to navigate our Canvas lab page, look through it just to kind of see how it's set up. You should have done a B data graph and submitted that on Canvas as well. You need to download the Excel app. If you have not done that and you don't know how, please email me or email IT or call them, figure out a way to get it on, because using the cloud version will not help you in this class. In fact, most of the assignments will not accept the cloud version, but most of the points, the bulk of your points of each assignment, cannot be done with the web version. They have to be done with the app. You already pay for the app. It's paid for in your fees, so you might as well download it because it's free. And then also an introduction Flipgrid video. So those Flipgrid, I'll give you the link to or a join code for a Flipgrid. Each section has its own Flipgrid. And so you should have made an introductory video. If you have not made that video yet, feel free to take this week to do it, but you'll have a different one next week to submit. If you did not do your B graphing data and you're worried about getting a 0, remember that I do not take late assignments at all. There's no excuse late policy across all sections. But there is one dropped assignment. So if this is your dropped assignment, then great. Just be on top of things from here on out. So what will we do in lab? We're going to apply concepts from biology while using scientific tools and methods. And I would argue This stuff should be reinforcing what you're learning in lecture. You don't actually get an end of the semester grade in this lab. 30% of your lecture grade is what you make in here. So that will be applied to your lecture grade. And therefore, if you're not seeing any parallels between what you're seeing in lecture versus lab, then you should email me and say, hey, what was I supposed to do with this? And how does it relate to lab? Or lecture, I'm sorry. How does lab relate to lecture? So we'll design and perform experiments. I don't think you guys actually get to design any since we're not meeting in person. You get the default ones that we give to the online students, but you do get to perform experiments. Some of them you get to collect data, but others you will be using data that was collected for you. And you will make graphs and a poster and potentially lab summaries to talk about what you learned. But most important, be creative, be fun. This class can be a lot more fun, and Dr. Shaw designs it so that even non-biology majors tend to have a lot of fun in this class. And usually whenever we're having fun, we're learning a whole lot more. So feel free to have fun with how you give your information back to me. Make your answers in your own real life. You know, if we're talking about photosynthesis, talk about photosynthesis in your own life, not necessarily the dry, boring side of science that sometimes, unfortunately, we see. So course policies, you don't get a handout. It's all online, obviously, but it's available on our lab Canvas page. Make sure you understand them and the procedures and our schedule. The schedule is online, but does not coincide with the schedule that you were given in the lecture. So make sure that you know that ours is different because you're in online labs. So what happens if you miss a lab? What is required for admission to lab? Oh, I went too far. So if you miss a lab, and obviously we're not going to lab, so you don't have to do anything to go to lab. But what happens if you miss a lab? Again, we have the lowest drop grade, so that's your default. No late assignments are done from there on out, but whenever you do miss a lab, make sure and contact me because obviously if you have COVID, then given that you have the OU online screening email, then all of those are dismissed and they aren't counted as absences. If you don't provide that though, if you miss a lab and you don't provide proof that it was from COVID, then it's just considered an absence as a normal absence. So like I said before, information, the syllabus it can be found on your lab lecture page. It's not going to be on any lab page, but our schedule is on our lab page. Again, lab information, you are either in section 39 or 40. Your TA is Shannon Reeves, and there's my information. And your due dates will almost always be on Canvas, due by 5 p.m. Now, remember that sometimes those things are due to change, and there might be a lot of assignments that say otherwise on Canvas. Canvas has like a ghost that comes and messes up all of our deadlines because this is such a large class of 1,500 people that Canvas doesn't like doing things correctly sometimes in that large of a class. So just make sure and follow any dates that are on papers and documents that we put on the Canvas page, not ones that are maybe promoted by Canvas itself. So what types of assignments are you going to complete in this class? So you'll have bonus pre-lab quizzes that are over the investigations that you'll be doing for the week. There are written summaries of the investigation and a graph of your data that you'll be doing, usually weekly. And you'll demonstrate that you know how to use the equipments and tools that we use in lab. Ravioli. Tips. Make sure and write these down. Do your pre-lab quizzes and background reading. So the pre-lab quizzes, I don't care. If you want to do them, they're bonus. If you don't want to, I don't care. You're going to be the same student to me. But if you're the difference between an A and a B, and at the end of the semester you come to me and you say, hey, I'm so close, is there any extra credit? I will say, yes. Go back in time and do all of those pre-lab quizzes, because that's really where you get your difference between an A and a B. I don't have the power to give extra assignments or extra credit or go back or grade anything different. The bonus comes from weekly little habitual bonus quizzes. So do those. Always do the background reading though, because background reading is really necessary for you to understand the assignment. Keep up with your online due dates. I'm not going to be sending out email reminders that things are closing or anything like that. There's just too many of you. And visit me in my office hours, either virtually or in person. I'm here to help you succeed. A lot of the time in online classes, it feels like you're kind of teaching yourself, and there's a lot of really big concepts, especially in lecture, that are going on here. I often participate in lecture and help teach in the falls, so I'm familiar with what's going on there if you have questions about concepts in lecture as well. All right, so what resources are available on Canvas? Again, I think we've been through a lot of this, but anything that you need that is going to tell you how to do something in our lab is going to be lab manual module. So how do I measure X? That will be there. How do I use a microscope? Although you guys won't be using microscopes. How do I write a lab summary? How do I cite something? Those will be things that are how-tos that are found in your lab manual. If you do your investigation modules, though, those are the week-to-week questions, like what are we doing this week? What topic is it over? All of that stuff will be your week-to-week modules. So all of this information, though, is fair game for all of the pre-lab bonus quizzes that you're doing. So be sure to make sure and know what you're doing each week and orientate yourself, navigate through the site, get to know what's available for you. All right, Flipgrid, what it is and how we use it. So here are your join codes. Flipgrid is just simply my way of being able to interact with you. It really helps me as a teacher know what you understand. So you'll be making weekly Flipgrid videos. This week's is just an introduction so I can get to know you about what you've learned. So essentially, what was hard for you? What do you still not get? What did you learn in the process? Stuff like that, so that my job as a teacher can be done, which is identifying where you lack in knowledge and helping fill those gaps. So Flipgrid is a good way. It's basically TikTok for education. Make a minute and a half video. You can use all the filters you want to. You can do a green screen on back. You can duet me. I don't care. Whatever you want to do to make this minute of biology explanation more fun for you, do it. I think you can do it in TikTok even if you want to, and upload a TikTok video, or do it on your iPhone, do it on your computer, do just a green screen of, or screenshot of you. Really, whatever you want to make this video so that I know that you know what you were supposed to learn this week. You'll create it to accompany all of the online investigations that we do. And again, this is kind of the summary of what we do. So section 40, your join code is 4021, section 40, year 21. Reeves 3921 for section 30. Reeves is on both of them. I'm sorry, I didn't say that. All right, so let's design a simple experiment to give you some practice. So you'll want to take some notes here. So this is an issue that's creating a lot of buzz among scientists. Ha ha ha, honeybees. So the USDA has noticed that honeybees are declining, and they noticed this this is December of 2016, so it's about 4 years ago in Louisiana. Researchers realized that the populations are decreasing and they don't know why. So we might come up with 3 reasons why we think that this could be happening. So this is essentially what researchers do. They find some sort of data that's interesting. Doesn't have a clear explanation on why it's going. It's some sort of phenomenon. And now we ask ourselves questions on why it could be happening. And that's how we come up with research questions. So 3 potential factors for why they could be in decline could be the introduction of a new predator. So maybe you've heard about those murder hornets that are going around. That's a new predator that's probably going to play a big role in how other species interact. So some of you may have heard of like introducing the mongooses to Hawaii a while ago was a public health measure that a lot of people did to keep other species that spread disease in check. But that completely changed the ecosystem of Hawaii. And if anybody knows anything about mongooses, they're kind of frightening. And so it also changed how people interacted with these animals. So introducing a new predator or just any new species to a new environment can often change the way those species go from day to day. So several years of hot, dry weather making flowers unavailable. We know that global warming, man-made global warming, is a really big issue and that things are changing all around the world. So maybe their food supply is going short or something like that. Or it could be environmental toxins, so maybe pesticides, something along those lines that are either man-made or maybe like we have a lot of methane gas stuff that's going on because of certain populations or needs for food. Maybe it could be something like that. But whenever we're researching and trying to come up with a good hypothesis, a good causal statement for why this is happening, we need to kind of explore and come up with a research question. Oh, another one, parasites and disease. And human disturbance. Okay, so there's more than 3. Human disturbance of bee foraging sites. These are all good guesses for why it could be happening. But it turns out it was pesticides. So new evidence shows a strong case for ban on chemicals linked to bird and bee deaths. So a lot of bee deaths, they assume, is caused by a neonicotinoid that's inside of these pesticides. So people are calling for them to be banned. This is something that is derived from nicotine, and throughout the semester you'll learn that these affect action potentials in the central nervous system. And if you're messing with action potentials, then you could potentially cause either seizures or paralysis, depending on what you do with those action potentials. And so in this, it's causing a decline in mortality among bees. So from here, scientists would go, okay, so how do we tell this is actually due to the pesticides? Well, we set an experiment up. So we asked the question, why are the bee populations decreasing? It's a good research question. We could test the hypothesis that exposure to neonicotinoids is causing the bee population to decrease. It's a pretty good hypothesis. Now notice this hypothesis has a causal explanation. Down here, a lot of people read this as causal. It's not causal, it's causal. That means it has a because or an explanation of why they think this could be happening. So, a statement that establishes a cause for the observed phenomenon. So if I were to say, do an experiment on why I think that sugar causes me to gain weight. If I say that it's because they're growing Cheetos on the moon, then I clearly can't come up with a really good causal statement for why that affects me to gain weight. Probably because those two things aren't related, right? So right away, if you can't come up with a good causal explanation, a biological, a plausible reason why this could be happening, that might be a suggestion that that is not sound. This is why, flat earthers are proven wrong very quickly, or anti-vaxxers, is because they have no causal reason, and that has to be backed up with data. So now we live in a very crucial time when information and misinformation and disinformation are all equally available. So you have to be good little scientists and learners and know that you're getting credible information. And whenever you put out information, not only in your classes, but also generally in life, you need to be able to back those statements up with a good causal explanation. That's what your hypothesis is. So most of you may have done if-then statements in high school. That's a good high school definition. We're going to upgrade it because we're in college, and we need to ask or we need to answer a question with a causal statement. If-then statements, if bees are exposed to insecticides, then they will have lower body weights. It's more of a prediction. It doesn't tell us why. It doesn't give us information about why we really think that's happening. So the scientists ended up testing their hypothesis by measuring different populations of bees throughout the world. So each different location was a different set of bees, and they realized that if the weight of the bees was decreasing, then that usually indicated that they were sick and there were morbidity issues or maybe even mortality. But either way, it was a good indicator of health. So in these different places around, it wasn't around the world, it was in a single county, not around the world. So different locations in the county, and they noticed that some locations had higher residue concentrations than others of the neonicotinoids. So they know that they're getting different amounts of neonicotinoids depending on where they were to go in that county. So their dependent variable, which is the one that they are measuring, is what they're trying to find out. And in this case, they're trying to find the weight of the bees, because the weight will tell us whether the bees are sick or not. The independent variable is the manipulated variable, or the part of the experiment that is different between the sample groups. So we had 25 different locations. So our different locations and the differences between them, that would be our independent variable, the locations. So we're comparing different locations based off of their insecticide residue. So to keep in mind and get these not mixed up, the dependent variable is influenced by the independent variable. Here's a little picture that might help with that. So independent variable is the variable that the researcher changes. So if we're doing an experiment based off of plants, let's say all of our plants, we have a bunch of different plants that want to grow, then we add water, but maybe in some of them we add fertilizer and others we don't. We do the same plant, let's say they're all peas, but we add a different amount of fertilizer each time. So depending on how much fertilizer, independent variable, we use, dependent upon it, we'll see how our plants grow. So growth or height of the plants would be our dependent variable. So our dependent and our independent variables go in specific places on our graph, if you didn't know that. I don't think I knew that prior to this type of a biology class in college. So making a chart or a graph is an essential part. It's the visual part that tells us essentially, here's what happened. Here's a pattern of all of our data. And on the x-axis, we will have our independent variable always down here. So this would be either our plant growth and then our fertilizer types. Opposite. Plant growth or our fertilizer amounts. Or if we're doing our bee data, then B, it would be the different places that we're going with the insecticides, the different locations and insecticide amount, and then based off of that, our bee weight. So if you're in Excel and you're new to Excel, just a little hot tip is that if you are going to put data in, then you'll want to use your columns as your groups, and your very leftmost column will be your independent variable for one group. The next column would be your next group. So we would have 25 columns right now, since we have 25 locations, all with data in each of those cells. So here are two types of graphs that we use often in this class. We use bar charts, which is the one on the left right here, or bar graphs. They're good for comparing groups or categories. So if I were to say how much sugar, we're doing a sugar and weight experiment, so how much weight will I gain? But we're doing people of different age groups. Then my groups would be people from 0 to 20, people from 20 to 40, people from 40 to 80, maybe 80 plus. Those would be different groups. Line charts are the one on the right. They're good for correlations or comparing changes over time. Keep in mind, you will not use this unless your independent variable is time. Now, we will use time as a tool in so many of our experiments throughout the semester. Very rarely, though, will time be our independent variable. So we will probably not use a lot of line graphs, but you might have done one of these two for your graph this week with the bee data. It might have looked something like this, but we don't really see a trend. And usually with a graph, you want to see a trend. So here's your line graph version of that data. And again, I'm not really sure what I'm supposed to be looking at here. So a scatter XY or scatter plot is what you should have chosen as the appropriate type of graph for your bee data. So this is it using a different experiment. So let's say that you all are students and you know that the more you study, usually the higher your grade is. So this is an experiment where each dot is a student, and it not only tells us how much they studied, so this individual studied 6 hours, but they also got an average of about what looks like a 25 on their grade. So these are used when both of our variables are continuous. So they're numbers and not a category. So categories like age groups. That's also a number, but you're in a group. But this, we're taking your actual score. We didn't ask you if you made an A or a B. We asked you what the actual value was. So because of that, and because the value we were looking for was study hours, then we have two continuous variables, and we'll use a scatter plot. You can see the trend that as people study more, so right here we have more hours, and over here we have higher grades, that as people studied more, they generally got higher grades, right? And as people studied less, they generally got lower grades. So that's what a scatter XY tends to look like. The trend line here will give us an idea about the relationship between the two variables. So like I said, the relationship would be, as I study more, I make higher grades. You'll be asked what the relationship is in all of your graphs from here on out, so it's important to be able to identify what the relationship between your two variables are. The equation is also something that we use to identify the strength and the slope, but it's kind of beyond the scope of this class, so you don't really need to know it. You just kind of have to put it up there if you do a scatter XY. So again, which of these variables depends on the other? How much we study or our quiz grade? Our quiz grade depends on how much we study, right? And maybe in the next week, how much we study will tell us, will be determined based off of our quiz score. So our final graph right here is called a box and whisker plot. We will also be using one of these. It's very similar to a bar graph. It has a lot of things in it, and it's good for comparing like a bar chart, but it gives us a little bit more information. So these are the whiskers right here. These things on the top, there's only one on the top here, and that has to do with how much data we have. But essentially it'll tell you what your maximum data was versus your minimum for that group. So if this is how much fertilizer we used, and we had one pea plant and we had one corn plant. So we used so the same amount of fertilizer. As we used more fertilizer, then our plant grew more. But it grew more with this plant than it did with this plant, because we can see that top, oh no, that top line back there, I can't go back. That top line back there told us the maximum data point. It grew the max that much in the min. So that's what box and whisker points or plots do that. If you have any questions about your 4 types of graphs and I'm going over your head on a lot of this information, don't worry. That's actually a lot of these students in this situation. And then about the other half are saying, I did all of this in high school. We have a wide spectrum of biologists in this class because it is an introductory class. So if you feel a little lost by this information, please reach out to me. I'm more than happy to go over all of this, but it is stuff going forward that you need to know and apply. So don't expect that you'll get to know it next week as we continue to use it more. You'll be expected to already have known it and then used it. So again, review, raviolis on the sheet. Here you go. Screenshot this, do something with it, write it all down, pause the video, write all this information, I don't care. But this is your graph Bible for this semester. If we are using a bar chart, it is because we're comparing, well, that says good for comparing. Good for comparing groups. If we're using a line graph, it's good for change over time, but only if our independent variable is time. Our box and whisker plot is good for comparing groups like a bar chart, but gives us more information. And then scatter plot is good when both variables are continuous data. So that should give you a good idea of the umbrella of graphs that we'll be using. So, your assignment. This week, watch the introductory PowerPoint video. Check, you've done it. Read the Keep It Cool Ice Experiment background. You'll need to know all of that in the PDF that's in your module in order to do your assignment. Make a graph from the data that's provided for you, and then a video on how to make a graph. Essentially, this video proves to me that you know how to make a video or a graph on Excel. If you don't know how to make a graph on Excel still, email me. We'll set up a time to figure this out together. If you are unsure to do certain elements in graph that are in Excel that are needed for your graph, email me. I'll tell you how to do that in Excel, or we can Zoom. But you're going to make a video essentially. It's about a minute long. I don't need a whole bunch of fanciness. I just need to know why you picked the graph you did, why it's good for your data, how you added your axes titles, if you needed a legend, how you added the legend, what your different boxes mean, stuff like that. So just tell me essentially how you go in and make a graph and what the graph means. But this should not be complicated, a minute and a half the most. You can either video screen, make a screenshot of you doing it and talking. You don't have to be visual if you don't want to show your face. Or you can do a screenshot of you talking like this with me. You can get your iPhone and simply video record yourself sitting in front of your computer and pointing to your laptop screen. I don't care how you do it. That is not what I care about. What I care is that you know how to make a graph and you can prove that to me within about a minute and a half. Lastly, you'll make your Flipgrid video if you have not, just an introductory video. And remember to use the join codes that I gave you previously. And lastly, I'm just going to go ahead and, oh, here is the Flipgrid stuff, the questions for the Flipgrid. This is all also on Flipgrid, so if you don't know what you're supposed to say in the video, it will always give you a prompt so that you know, and this is the kind of stuff that I am asking this week. But I'm always going to leave you with learning outcomes, and these are the things that I want you to walk out of our classroom having known. So you should understand the relationship between independent and dependent variables at the end of this week. You should be able to identify your independent and dependent variables, and you should be able to formulate a causal hypothesis. You should be able to understand your data and know how to compare it. You should be able to make a graph out of that data in Excel and tell me why the graph that you used is the appropriate type of graph. Lastly, how does this relate to lecture? So in lecture, you have probably learned how organisms maintain homeostasis and what things are really important to do that. You are probably going to learn this week surface area to volume ratio. How is that crucial to maintaining homeostasis? And how does that apply to the experiment that you're going to do today? So again, this information is not anything that I'm requiring you to write down or prove that you know, but it's what you should be walking away from this class knowing. So if you don't, then know that you are going to be tested on this stuff at some point in the semester, so you should probably figure it out, either with my help or if you want to on your own. So without that, if you have any other questions, feel free to email me. We can set up a time to meet in person, online, or we can just email the whole semester, whatever you're comfortable with. But know that no questions are stupid. But if you ask questions like, where can I find the syllabus, or when is this due, yours might not be the email that I reply to right away, since that information can be found somewhere else. Then I'm going to devote a lot of my time more to answering emails that need to be directed to students that need teaching. So if I don't reply to your email right away, that might be a hint that that information can be found somewhere else, and I have too many students to reply to that one. But always here if you have any questions, reach out, or if you just want to say hello, and I hope you guys have a great week.